Huge thank you to the committee for inviting me along today. These are often conversations that we have with our collaborators when we're trying to figure out, how to initiate a new project. And so it's been a really useful exercise for me to think about, how we communicate our science and what we're doing to folks that are going to start new projects with us and get down and dirty with dental calculus. So as was mentioned, I am at Penn State in the anthropology department, but I'm a biochemist and microbiologist by training. And so I really think about archaeology probably very differently, I think, than most people who are classically trained in archaeology. And one of the reasons that, I was kind of brought into archaeology is because I think a combination of microbiology and archaeology together can really just help illuminate so many of the sort of hidden variables and hidden information that we've been trying to recover in the archaeological record. So I'll be talking to you tonight a little bit about, kind of the background for ancient microbiome research and why I think it's really important for archaeology. And then I'm going to go into the practical specifics, like how do we sample dental calculus? What information should be recorded? What do we need to watch out for? And how do we do this, in a sustainable way? And then I'm going to hopefully share some of the why and some of the research behind this is why we have these recommendations and why they matter. Because we're trying to get out very rigorous, robust data from these very valuable samples from our ancient ancestors and people of the past. So I usually start out most of my archaeology talks by just illuminating the fact that we live in a microbial planet. Every surface that you touch, every bit of air that you breathe, every sip of water that you take is littered with microorganisms, and they underpin most of the basic processes that happen on the planet today. The human body is also no different, right? We carry tons of microbes in us, and we interact with that microbial world using sharing of our microorganisms. Now, we refer to these discrete microbial communities in the body as microbiota, but it could also be a discrete microbial community in the soil or on an artifact or whatever that looks like. And it just means a collection or a community of bacteria, fungi, viruses, any small type of living organism that live in an ecology sort of together. And to give you the scale of this, if you just think about your own body right now, wherever it is that you're sitting, you have over 100 trillion bacterial cells in your body right now. In fact, that equates to more than half of the total cells in your body. So humans aren't really just human, we're kind of a host for microorganisms. This equates to over 1,000 species. In fact, in your mouth right now, you probably have about 200 different species. And collectively, this weighs 1.4 kilograms of your body weight, which is analogous to your brain. So you're carrying around an extra organ's worth of weight of these microorganisms, and they underpin a lot of really critical processes for your body. And being an ancient DNA researcher, we're able to kind of reconstruct these microbial communities using their DNA. And so when we think about all the functional and genetic content that those microbes bring along with them, we refer to that as the microbiome. Now, if we think about the genetic content of these microbes that live within the body, the genetic content outnumbers human DNA over 100 to 1, because every single person has somewhere between 2 and 5 million bacterial genes. So that means 99% of the DNA in your body is actually microbial. So if we want to understand how humans evolved, if we want to understand what makes us human and how we adapted and interact with other people in our environments and animals, we have to be able to trace and to track the other 99% of DNA within the human body. And these microorganisms are not static. These microbial communities change and evolve over time, but they also adapt to different pressures. They're subject to selection. And these processes can happen in really rapid timeframes, in the matter of a single exposure or, something very, very brief and very rapid. But they can also have, some longer term periodicity and change with them, which means that how microbiomes change and adapt through time and space can be used as proxies to understand many of our questions in archaeology that remain difficult to access. We can, use microbes as proxies for diet. Because microbes eat the same thing we eat, we can look at their functions and what they're sort of doing, and that might be a proxy for the types of foods that we eat. Because microbes vary according to different environments, we might be able to understand where people were and what sort of context they were living by the microbes that they shared with that environment. Things in your daily life, your exposure, things like jobs and chemical exposures, and who you interact with can also mean that microbes from those environments are imparted on your body, but also if you're exposed to things like chemicals, bacteria in your body can respond to those. Toxins affect microbes similarly to the human body, or sometimes in different ways. And we can use those as proxies of all these exposures. Humans and pets and non-pet animals share lots of microorganisms. In fact, you can tell whose dog is whose based on the shared microbes between that dog and their owner. So we can really track then were people exposed to different animals or living closely with them. And we can use microbes to track medical treatment. Was somebody receiving antimicrobials or things that impact bacteria? And same thing, we can see, shifts in sort of pollution and long-term exposures, and even shifts in the microbiome linked to seasonality. So microbes serve this really critical role in archaeology because they're a proxy for understanding many of the biological mechanisms that we're trying to trace. So there's a couple different ways to study ancient human microbiomes. One of the ways that came out, straight away, and this is kind of 2012, was, using coprolites. And, this was really exciting. We thought, oh gosh, like there's so much done in the gut microbiome work. The trillions of microbes that live in your gut and help you digest your food and are responsive to your immune system. We really thought that this would be an awesome avenue to explore, to be able to reconstruct the types of microbial communities that lived in the human body. Unfortunately, though, for us, this was a very difficult process because of several different things. The first one is that coprolites are littered with environmental DNA contamination. Wherever that piece of feces was initially planted, it then starts to interact with that sort of soil, for example, and many of those organisms that live in the soil now are part of the feces. And so distinguishing what's contamination and what's endogenous to the human become very difficult. The other issue is that the microbes, that come out in feces are still active even after they're deposited. And so, they continue digesting some of the leftover nutrition in the feces. And what that can mean is that the microbial community will shift in its composition and start to look more like the microbes that live in a compost pile rather than the microbes that live within the human gut. So while coprolites can still be really useful in, say, tracking individual strains that might have still survived, or being able to do genomic analysis comparing two different bacteria, it's been very difficult to use them for microbial community analysis or whole microbiome analysis because of some of these complex issues. So lucky for us, there is another sample type that's been, incredibly useful in us reconstructing microbiomes of the past, and that is dental calculus, which you're all here to think about tonight. One of my favorite sample types. And this is just preserved calcified dental plaque. And often I think people in the UK will call this tartar. This is what your dentist removes with the little sonication or the scaler, in your mouth. But in people of the past, right, they didn't have modern dentistry to remove dental calculus. And so it built up over the individual's lifetime. And in fact, calculus usually starts forming in sort of early adulthood. We think of this being about, somebody who's the age of 18, and then continues to form throughout someone's life. And we think that its formation can probably vary over time, depending on your diet. You do have to have carbohydrates in your diet to get calculus to form, although it still forms in people even without some of that, just at lower levels. And it really traps in place microbes that are endogenous to the mouth. So as you sleep, the pH in your mouth changes and calcium ions out of your saliva precipitate and turn dental plaque into, a soft, removable substance into a rock-hard cement-like matrix. And that cement-like matrix, really traps those microorganisms in place, which is excellent for us because it means that, those microbes were really put in there in place while someone was living. It also means because they're in a rock hard matrix, we can do things like decontamination. We can remove some of the environmental outer signatures and know that what's inside is still from that individual. One of the other nice things too is that calculus also tracks specifically to an individual because it's on top of a tooth, whereas something like a coprolite, you don't know if it's the same person from multiple coprolites or you're looking at a population level analysis or a single person. Whereas calculus, we can trace directly to the individual. So we can match the microbiome data that we have alongside, oral health, systemic health, any sort of individual, health records that may be possible. So dental calculus has absolutely transformed the way that we're able to reconstruct and to think about ancient human microbiomes. And my team has been incredibly excited about the promise of calculus for archaeology, but also the promise of using dental calculus to retrace and understand the origins of chronic disease. We think that this is, truly illuminating for modern human health, as well as it is for understanding the past. So I think when we started publishing some of the first dental calculus papers in 2013, folks were really excited not just about the bacteria, but about all of the things that get contained into calculus. And so I'll talk a lot tonight about bacteria. Again, I'm a microbiologist by training, and so that's my love and my passion. But it's important to note that dental calculus contains all sorts of additional proxy information. And so things like human proteins and bacteriological proteins have been removed from dental calculus. Some viruses have been found in calculus, although they're usually viruses that infect bacteria. They're what we call bacteriophages. And then again, there are some particles from food, as well as other bits and pieces that can be used in the mouth. So ropes or toothbrushes or whatever that looks like. Some of that can get stuck in dental calculus and recovered over time and space. So it's not just that the bacteria there. I argue that they're probably the more useful subset of information, but there's lots of other additional sort of what we call byproducts of our analysis that can be incredibly useful in archaeological contexts. I will say, though, that bacteria are usually greater than 95% of the DNA that's pulled out of dental calculus. So if we're doing what we call a metagenomic approach, a multiple genome approach, 95% of what we look at are going to be bacterial or archaeal genomes, these microorganisms. A very small proportion of it will be human DNA, a very small proportion of it will be, food DNA, and a small proportion of it too are maybe human-associated viruses or bacteriophages. So really identifying how to use the bacteria in unique ways ends up becoming what we get really excited about because it's the bulk of what we recover from ancient dental calculus. So very early on, Keith Dobney, who was a student of Don Brothwell, who some of you maybe on the call might know, was really instrumental in getting us to work on dental calculus. And for many years, he got some ancient DNA scientists to try to recover ancient DNA from dental calculus. And it was simply very difficult to do because reagents from molecular biology were too contaminated with bacterial and microbial DNA to be able to identify the signatures that people were looking for. And with the revolution of the Human Microbiome Project in the United States, which happened around 2010, producers realized they needed to clean up their reagents, which then opened all sorts of new avenues for recovering microbiome information and bacterial information from dental calculus. But prior to that stage, people were really using microscopy to try to figure out, what was in dental calculus and kind of what it was made out of. And this was published in the supplementary information of one of our first papers applying high throughput DNA sequencing to calculus. And you can see this sort of picture here of the maxillae with the teeth here. And this is actually a huge piece of dental calculus. So I really think about how much pain this person would have been in, to have this happening to them. But this allowed us to be able to remove this and do some really high resolution microscopy imaging on it. Here you can see the formation happening in layers of the dental calculus. So it's a bit like tree rings. And, supposedly there were some individuals of Jewish descent where they could see kind of this happening in kind of layers of 7, and that the 7th one, maybe the day of the Sabbath, was much larger than the other rings. But certainly we have rings like a tree, and we think it kind of grows out from probably some sort of base formation, and then there's layering on top of that. I will say that there's a lot of research that needs to happen in how this forms and the different types of microbes and the different layers and its formation, but that's an area that we're actively still working on. If you zoom in even further, they were able to see these sort of spheres. These are cocci. These are microorganisms that are very common in the mouth. They're what we usually call streptococcus. And usually when I say streptococcus, people think strep throat. And you're right, it's the same genus, but you have lots of oral streptococci that are very beneficial in your mouth. And so this is really confirmatory that we were able to retain microorganisms preserved and locked in place in dental calculus, which was really exciting for us. And Christina Warner's group also did some really amazing microscopy of dental calculus. And here it's a side slice, but you can see that calculus hanging off. And again, able to show even more diversity of microorganisms preserved within this. So again, it's largely a bacterial biofilm. So the majority of the signatures you're going to get out are microbes that are endogenous to the mouth, that like living in the mouth and live in the mouth when the person is healthy and going about their daily life. There is some pathogen DNA that can be detected from calculus, but it is really limited. I think, when we started the field, we were really wide-eyed and bushy-tailed and thought, gosh, we'll be able to record, any sort of oral respiratory pathogen. And you can pick up pieces of those, absolutely, for sure, but it's much less abundant than we would have initially anticipated. So I wanted to just insert this quickly and show you that this is what this looks like in living people today. So it's not just that there's two, spheres and a couple rods, and that's what it looks like. This is actually some images taken from a lab at Harvard, the Broad Institute, Jessica Mark Welch. And there's just incredible diversity trapped within this calculus and trapped within this. So it's not that we're just looking at one or two species. We're actually looking usually at over 200 different species. And we're looking at them in very ecological contexts. These microbes form forests and valleys and, have larger ecological roles that they play with one another. They're antagonistic to some, they're beneficial to others. And so these microbes are really living in their own niche and their own world right there on the tooth surface. So it's exciting that we're able to now capture this and reconstruct it. So I want to shift gears a little bit to now the practicalities of how you would go about, getting a dental calculus sample and then, doing some ancient DNA research on it. And so I thought I would start off with just kind of the process, the broad process, and think about, if you're going to do a dental calculus study, what are the things that you kind of need to be aware of and think about in the context of information about an individual before you would even say, yes, let's go ahead and collect dental calculus from that person and include it in our study. And so one of the biggest things that we have been working on, and that has really kind of upended the field recently, is thinking about what we call oral biogeography. And this simply means that, there is a micro scale of different geographies, different ecologies within your mouth for these microorganisms. And so, this lovely cartoon, which you saw earlier in the talk, illustrates some of these differences, but not all. And some of the differences that were well appreciated, I would say, even in the archaeological literature, were the difference between what's called supra or above gingival dental calculus versus subgingival or sort of below this cemento-enamel junction. And there was quite a bit of confusion. I would say it maybe still exists in the literature about what is a supra or a subgingival dental calculus sample. But most of the work, in fact, the majority of the work that's been sequenced today in the literature is all on supragingival calculus. And this is what it looks like in an individual actually from the UK during the medieval period. And we use this picture a lot to help describe, what we mean when we say supragingival calculus. In this particular individual who, doesn't have periodontal disease, you can almost imagine where the gum line would have sat, and you can picture this dental calculus, sitting really on top of that gum line. It's really above the sort of gums, surface, and it builds up. And this is quite a ledge here. So it's something that sticks out, in a measurable, observable distance because that biofilm is literally sitting on top of the soft tissue when that person is alive. So subgingival calculus is something that is on the root surface way down here. And in somebody who has periodontal disease, where they've seen this alveolar bone loss, you do have some exposure potentially even to these roots. I can imagine the pain that person would have been in, but you can get dental calculus that grows down here. Quite often it's paper thin. It can often be green in color because it contains different microbes. And it's very visually distinct compared to what we consider supragingival calculus. So if you have individuals that have, like paper thin type calculus on the roots of the teeth, you can sequence that as dental calculus, but it might tell you something very different than all of the samples that have been sequenced before up here, that were exposed to the mouth while someone was sort of living. So being aware of even what, major type of dental calculus you have to begin with, I think is really important. The other thing in the context of oral biogeography is just which tooth this dental calculus is found on. And the short of it is that you have different microbes that grow in the front of your mouth versus different microbes that grow in the back of your mouth. And so, you have different oxygenation in the front of your mouth. As I'm sitting here talking, you can, it's open to the air, right? It has different oxygenation levels versus in the back of your mouth. These are more likely to be sort of anaerobic communities sometimes. The sort of access to saliva is very different near kind of these salivary glands in the back of your mouth. So there are just different types of microbes that grow in these different ecologies. So even knowing that you can collect dental calculus from the same tooth across your individuals becomes really, really critical. And we looked at this in a paper we published with the Museum of London folks, and it was a really a labor of love project. It was just awesome. But it includes over 127 people from the Museum of London, as well as a wide range of other people across Britain. And this was the first time we were really able to show this sort of effect of which tooth you sample calculus from drives the majority of variation in data sets. So these are plots. This is a principal components of analysis plot where every single dot represents an entire bacterial community found in somebody's mouth. So the closer the dots are, the more similar the communities, the further the dots are apart from one another, the more different the communities. And what you can see is that most of the incisors cluster over here, whereas most of the molars cluster over here. Your bicuspids, kind of are in the middle here. But simply depending on where you pull your calculus from can drive some of the signal in your data set. We also looked at, other oral biogeographic factors, things like tooth surface, the right or left side of the mouth, mandibular versus maxillary sites. And I will say, depending on the population, depending on the types of microbes in people's mouths, we have seen effects of all of these in different data sets. But the really critical ones, I would say, are which tooth it's coming from and whether or not it's supra or subgingival calculus. Tooth surface, we think, is also quite important, especially in this British study. We were able to show a significant difference between buccally sampled calculus of the same tooth versus lingually sampled calculus from the same tooth. So again, that's largely driven probably by proximity to salivary glands and different types of nutrients. But being aware of where you're able to even remove calculus from, and is that similar across all the individuals you want to compare and contrast, is really critical. And there's been a couple of calculus papers that have been published where they have, mixed calculus samples from different parts of the mouth. And there's different amounts of mixing in different people. And they've failed to find differences across individuals. But part of what they've done is just absolutely increase the noise in these data sets and lost the ability to really accurately compare and contrast across individuals because they can't account for oral biogeography. So this is kind of our number one, big, soapbox to get on right now, is that if you're going to start a dental calculus study, you have to collect information about which tooth it's coming from, and you have to, be aware of its sub or supra, and probably even absolutely also collect buccal versus lingual. And if you have random calculus from different people, say one person has an incisor, another person has a premolar, another person has a molar, you might want to think about what you can do with that data versus what you can't do because you don't have samples from the same tooth. I think too, just thinking about, our collection, we do this from sort of one tooth at a time because we really care about that oral biogeographic information so we can compare and contrast across people. And that really also ensures that this is a much more sustainable process over time, right? We're removing one sample off of someone's mouth rather than removing all of the calculus off of all of the teeth, which can be even more destructive than, the research already is. And so, also just thinking about your sampling strategy in the context of conservation and in the context of sustainability, I think is really important. And a couple of years ago, Abby Gans, who also led the large British study I just mentioned, did a survey during the middle of COVID when we couldn't be in our labs and couldn't be doing all the fun things that we wanted to do, led a survey of dental calculus researchers and asked them, how do you sample? How many teeth do you collect? What are you thinking about? And in fact, it was a pretty big variety. There were about 20% of us that collected off one tooth and one tooth only. There were, almost a, a slightly increased number that collected 2 teeth. But there were some people that collected calculus off of, 3 to 5 teeth per individual or all available teeth. And I'm really concerned about the destructive nature of this type of sampling that happens, because there's certainly enough DNA within one, calculus sample from one tooth, even though it's very small. Our rule of thumb is if it can be picked up with a set of tweezers, we've got a sample that's probably big enough to work with. So, just really, really thinking about that procedure and thinking about sustainability, I think is really, really important. But in addition to recording oral geography and thinking about that, you also want to think about additional information that will help you interpret the data that you have in hand. And so other information that we think is really critical is oral health. We know today that people who have, different oral diseases have different oral microbiomes because of those diseases, or, were maybe even caused by those different bacteria. It's kind of a chicken and the egg thing. So what you really need to, have a handle on too are the oral diseases that person was suffering from. And so we really appreciate some bioarc, support, I think, in our studies where we can record, presence of caries or cavities, presence of periodontal disease, abscesses, tooth loss, LEH, the list continues. But anything that's considered kind of an oral health issue, it's really critical that we have some bit of that information. Because if you're comparing people who do and don't have periodontal disease, you might see differences across them, not because of their lifestyle or their diet or things that they're doing in their daily life, because they are suffering from differential disease. And so you have to be able to control for disease before you can get on to these additional, cool things that I think microbes can tell us about archaeology. We're also interested in as much information as we can wrangle. So, systemic health was a big part of that British paper and has gone on to be a big part of our research moving forward, and trying to understand how oral microbiomes are linked to, all of the sort of systemic indicators of health and disease that you may be able to record using bioarc methodologies. Things like sex, age at death, archaeological age, cultural affiliation, burial context, like whatever information you can glean about that individual, it's going to help us interpret, why they have microbes that they have, and what those microbes might mean. Really critically in this paper where we surveyed different calculus researchers, we also found that what was not being recorded was the methods in which people were using to record all of this information. And so, great that people were recording whether or not folks had periodontal disease, but if you're using two completely different bioarchaeological methods to do that, you might have had two very different classifications, and those two aren't comparable, or that you can't interpret your data in the context of two different methods. So it's not even just recording the information, it's also including your method in which you use to record that information at the same time. And that will, make sure that you can interact, that your data that you're generating from this individual can be incorporated with lots of other data sets and lots of other studies, which is ultimately, where you want to head to be able to compare your person of interest to all the other information that exists now in the world. So if you're new to this, I will point you to the Gans et al. AJBA, American Journal of Biological Anthropology, paper from 2024. We do have SI documents for archaeological dental calculus recording guidelines that can help point you in the right direction on how to collect all this data and record it. We had also, with the paper, submitted an Excel sheet, which has what we consider the baseline information that you absolutely have to do dental calculus analysis moving forward. And you'll notice a lot of this is pretty, standard type museum information and where the person was buried and who's collected it. But we want some basic oral biogeography information you'll see here. So, tooth number, surface, etc. We then have, information here for sampling notes and photography of the individuals. We recommend that people take pictures and document the teeth before dental calculus is removed. There's some basic information about sex and age. And then oral disease being sort of really critical. Now you can add things onto this, but this is what we effectively have to have if we're going to be able to interpret the data in meaningful ways in the context of archaeology. So if you want this sheet, feel free to shoot me an email. I'm happy to email it out to anybody. It was supposed to be incorporated in the paper and they forgot it in the SI documents. And we've emailed the journal and they're supposed to put it up, but they still haven't yet. So anyway, if you need it, let me know. But this SI document is in place that kind of explains the process. So once you've, I say like identified, the individuals and that you can collect data on them or you've got data in hand, then you can move forward with sampling, and only after you kind of understand that. So, our sampling process is not rocket science. It's actually pretty straightforward. But I think it's also important to get right, so that you don't damage the tooth surface of the individual where you're sampling, and also that, you don't have mishaps. So, we've definitely collected dental calculus before and had it go ping, and fly across the room, and then everybody had to freeze and walk on eggshells until we could find it and pick it up and bring it back, right? So, so these sort of, ideologies have really been established as ways to just make this process as easy as possible. So we have what we call our ancient DNA sampling kit, and this is generally what we kind of have in it. We've got aluminum foil, we've got a dental pick or a screwdriver. Some people like using dental picks with this sort of curvature. These dental picks, though, they're designed to flex, to be used in the human mouth, when someone's poking around your mouth, and not be super hard and rigid and cause damage to your soft tissue. And so we actually did a little bit of testing and found that there's this one particular screwdriver type pick pack that comes from Craftsman. And, there's a couple other companies that make kind of knockoff ones, but these sort of curved pick type tools are rigid. This is, a harder steel. And these can even sometimes work better for calculus, depending on what you're trying to remove. But a very fine point, and just something that's potentially more robust. Some of these can be quite flexible and can be problematic when you're trying to sample. We always encourage that people use PPE or, personal protective equipment. There's two L's there. I was going to say minimally. This needs to include at least gloves and a face mask. So just a surgical type mask. And the reason we say that is because as you're talking, you are spraying oral microbes everywhere, and you're not intending to. But if you've plucked a piece of calculus off of somebody and the back side of that was on the tooth surface, it's effectively naive. And if you're talking over top of it, you're just spraying and contaminating it with oral microbes. And so, the face mask becomes really critical while you're sampling dental calculus to avoid you imparting your oral microbiome on top of that dental calculus specimen. We also have a lot of gear like 3% bleach, 90% ethanol for cleaning tools in between the process, wiping down any benches or areas where we might, be working. And then we've got sterile bags for collection of the calculus. People used to collect them in tubes, and the static electricity between calculus and a tube is a nightmare. You can't get it out. It gets stuck in there. And so everybody's moved to bags. So please use plastic bags. You need a marker and towels. Good golly, not marker. That, you need to clean and to record and to document. So after you've done all your recording, after you've taken your photograph, then you want to clean the area with bleach followed by ethanol. The bleach actually removes the DNA and breaks it down. The ethanol just removes the bleach so that the bleach doesn't get on your samples and on all the things that you're working on. You then identify, your single tooth. And then, again, great if you've already photographed and recorded, but just really hitting this home, like you have to photograph and record. So if you want to do it here or before, up to you, but like that's your last call. Like you have to have it done before you do that, because the next step is to literally remove it. We usually lay out a large piece of foil, probably, I don't know, 30 centimeters, long. But it'll depend on the individual. And then we wrap that foil around the area where we're trying to sample. So, if the calculus is here, we're going to wrap that foil around, so we can still see where we're going. But if the calculus flies off, it's going to get caught in that foil and retained. We then identify that sort of ledge for supragingival calculus, where you've got this, where you could have imagined the gum line being, right? This is a ledge, and that's where we're going to apply pressure with our dental pick. So, we would take the sort of edge of this and we would push, and ideally if that mandible's upside down, like that's even better. So you want that ridge to be facing up at you and you're going to push down on that ridge. And by pushing down on that ridge, it's going to cause the calculus to fall off the tooth surface. It should pop off the surface of the tooth. And it might not mean the whole piece from that side will come off, but at least a large chunk of it should come off together. if you push on that and your instrument goes all the way through and kind of digs a valley or digs a crater, often what that means is that the calculus is not well enough preserved to have maintained its structural integrity. And you're probably going to have preservation issues. Not always, but probably. So it should, pop freshly off the surface of the tooth, and it should come off in a chunk. Again, we're looking for chunks that are big enough to grab with a pair of tweezers. Those can be as small as, say, 2 millimeters by 2 millimeters. They can be really tiny, but they better be in chunks and not sand grains. Again, if you've got sand, you've got big problems because you can't decontaminate in the next step, which I'll talk about in a second. So hopefully you've got chunks, and then hopefully it's fallen off, into your foil. You can remove the mandible or skull, like whatever it is that you're sampling from, pick that foil up, kind of fold it into a valley, and pour that into your sterile bag. We really, the sterile bag, this has to be labeled with critical information. So, museum number, who sampled it, what date, where it's from, and maybe even some, critical additional descriptive information like oral geography, so that you know exactly, what sample it is and where it came from. After you're done with that, you then need to clean your area again, swap out your gloves, as well as clean your implements before you start with the next individual. And so really trying to avoid, cross-contamination during the sampling process is really important. And again, removing your gloves and changing those out is really important. The mask can just stay on the whole time. You don't have to worry about taking that on and off. Just keep it on and keep it, over your nose and mouth, properly worn. And you should be sort of ready to go. I will say, we've also worn full suits to sample. If you've done other ancient DNA work, you might have seen people in full Tyvek suits. We occasionally will wear those. Usually if we're sampling in a museum, though, where individuals have been stored for, decades, often we'll just wear long sleeve shirts and, our gloves and our face mask. And, the thought process is there that these individuals have been handled by many folks, right? They are just going to be fully contaminated on the outside. And so, as long as we're not sloughing skin cells onto them directly, we're doing sort of as much as is necessary as needed to do. And we've seen, good results from sort of that process. But certainly gloves and face masks really critical. We did publish a paper with Bastian Llamas many years ago that kind of walked through some of the thought process here and some of the thinking on lots of ancient samples, but including calculus. And we've also published a paper on trying to, make this method a bit more streamlined and a bit more obvious, if you want to sort of check that out as well. So after we sort of collect dental calculus and it's in these bags, we then will, ship it to a specialized facility. So, it needs to go to an ancient DNA facility. I would say, ancient DNA on microbes is the craziest form of ancient DNA, especially, human-associated microbes that are also present in the technician and everybody else that's working, that they're constantly spraying as they're talking about. And so your methods in your ancient DNA lab of decontamination, of making sure everything is truly clean before you work, just have to be absolutely up to snuff. And that's a whole nother lecture in and of itself. So if you can't just process these in normal old lab, and even some clean labs, even some ancient DNA labs may not be well spec'd to do ancient microbiome work. Even if it works well for ancient human DNA, it might not work for ancient microbiome DNA. Microbial organisms are everywhere, and so you've got to be able to control for that. Largely, again, we'll just extract DNA into a tube, we'll do some DNA sequencing, and we'll do bioinformatics analysis to sort out what types of bacteria are there, what abundance those bacteria in, and do some genome assembly and some functional analysis to figure out what are those bacteria actually doing. So I want to spend just the last few minutes of this talk, providing some information about why we care so much about, these methods that we've put in place, and some of the research we've done to really improve on this methodology to make sure that, the way we sample calculus and the way that we analyze it really is trying to get at the true, oral microbiome that was present in these individuals, and isn't just the result of contamination or of, a modern technician breathing and talking over our sample, and really trying to ensure that what we're looking at is real. So there's a couple really key impacts here that I think you really need to be aware of as somebody who might be interested in doing calculus research. And that's just thinking about how the DNA that you're going to look at from ancient calculus specimens is going to be fragmented and small, and it's going to be damaged. And we initially thought this would be a big problem in analysis. It turns out it's not. And it actually ends up being a good thing because it allows us to authenticate the fact that we're looking at ancient oral microbes and not the technicians' oral microbes, or even archaeologists' oral microbiome. Or, we were talking just before this about archaeologists that would lick bone versus rocks to figure them out, right? And so it's very possible that we have old oral microbiomes on there from 50 or 60 years ago from an archaeologist or from someone else, right? So that damage becomes really critical for us to suss that out. We're really, concerned about contamination and making sure that modern bacteria from the world around us, from the technician, from the archaeologist, from everybody else, from the soil, that's accounted for and gone. And we're really trying to get a handle on, how we account for microbes kind of degrading over time and space, potentially at different rates. So I'll fly through some of this, but I'm happy to ask answer any more questions that you might have. So again, our DNA is going to be very, very fragmented. And we did a lot of work to figure out whether or not, the size of that DNA would sort of impact whether or not we're able to recover communities. And the answer is that it does impact things. And there are some species that we just miss altogether if we have sort of, really short pieces of DNA. But at the same time, there are swaths of the oral microbial community where we're able to get a really good handle on the types of taxa that should be present. And you'll see those same colors here as, sort of shown here. It doesn't mean that we recover everything though. And so, the absence of evidence is not the evidence of absence. We have to be careful in how we interpret the results because it might not be there because of taphonomy or because of the short pieces of DNA, not because it's actually not there. So including fragment size in any analysis you do, and understanding how old your samples are and how short you might expect those pieces of DNA to be can really go a long way for helping you figure that out. And, as you're talking to ancient DNA researchers about what they're going to do with these samples, it's really important to ask them, what methods are you going to employ here? What are you going to use? And are you going to be consistent about, how you employ those methods and what data you're going to compare those methods to? And in fact, different types of DNA extractions, these are two different extractions that are used very commonly in the ancient DNA field, will pull out very different lengths of DNA from samples and will also, enrich or miss certain species depending on which approach is used. So certainly make sure that, you're assessing what methods are being used and that those are consistent across your sort of efforts. And like I said before, the short pieces of DNA, they can be more, frustrating to work with, but we're still able to accurately really assess what's there. And these short pieces can really be indicative of ancient signatures. So we can really be sure that we're looking at ancient DNA rather than sort of our technician's DNA. The second key thing is just contamination. Again, many of the people that we study today were buried in the ground. And so, we're thinking about decomposition microbiomes. We're thinking about soil microbiomes. We're thinking about the archaeologist's oral microbiome, all this stuff that could be on the outside of that dental calculus. And so we have to figure out ways of decontaminating samples and of dealing with that contamination. There's also been a lot of talk in the field about leaching and whether or not DNA from the environment can actually get inside of calculus and what that might mean. But there's still lots of work to do in this space. I would say though that we've come a long way. And so this is an example of that, right? So if your true oral microbiome is really here in orange and blue, but you have, for example, samples processed in different labs or samples that came from different sites, something along that, you might have different types of contaminant DNA. And you might have two samples that should look exactly the same from their endogenous type, but actually show up very differently in your analysis because that contaminant signal is contributing to the overall signature of microbes. So being able to include contamination is really important. And one of the ways that we do this is by including controls in our analysis. So if you are lucky enough to be an archaeologist that's actively excavating and you're going to sample dental calculus in the field to send to a collaborator, you should also be thinking about what sort of controls you can collect at the same time to really improve that analysis. And so what we've done now is think about soil samples that are directly adjacent to the person or to the individual, potentially from the back of the skull, something like that, that's not directly associated with the mouth, but somewhere we can get a good signature on the soil microbes that might have been present. We'll also do swabs of the museum, swabs of the box the person was in, or whatever that looks like for you. But just trying to track, what are the microbes that could have been kind of in the exterior, or, as part of that contaminant profile that you might want to look at. We also include controls from our lab environments. So even our crazy ancient DNA labs, where we're in full Tyvek suits, it looks like an Ebola lab, right? There's still microbial DNA present. And so you have to include what we call extraction blank controls. You've got to include no template amplification controls to, again, make sure that what you're seeing is truly linked to that ancient person and not to how the sample was processed and where it was processed. And so this is, again, just reiterating, these ultra clean labs. And I often use an Ebola lab as an example, but it's really just making sure people understand that you're trying to protect the sample from yourself rather than protect yourself from the sample. In an Ebola case, it's reverse here, but many of the ideologies are the same. And again, different methods have different control profiles. So important that you constantly monitor that and don't just monitor it once and call it a good, good deal. That's really critical. The last thing is just how you decontaminate a sample. And so Andrew Farr, who did a lot of work with the Museum of London, and he's at the Cambridge Science Centre now as a science communicator, did a lot of work in my team thinking about this and really using different treatments of EDTA, bleach, UV, to figure out how we get the best decontamination or removal of those contaminant sequences from calculus. And we're able to do a pretty good job of removing environmental taxa and increasing oral signatures through some of those methods. Lastly, I will just say how you store your calculus also matters. So if you're a museum curator or an archaeologist and you're thinking about storage in between the times, cold is better, right? So if we can get ancient samples in freezers as fast as possible, that's where we'd like to head, rather than room temp storage or the back of someone's van or truck storage. So keeping these things cold is going to preserve their DNA a lot better than sort of letting them get warm. And you can see that here in this plot. These frozen ones look a bit more similar compared to these room temperature ones, where we now have seen blooms of particular taxa and can really alter the results of the study. So that's it for me today. I'll leave you with a couple questions. One is just, how might microbiome science add to your archaeological research? Can it help you with diets, migrations, health, lifestyle? What do you want to know? Because I bet microbes can contribute some new information in some way. And I hope you'll think about, what pitfalls might you encounter during sampling or during microbiome analysis, that you as an archaeologist might be able to contribute to the conversation and making sure researchers, are doing things in the best way possible. I hope if you take away anything from the talk today, it's that calculus is an amazing resource for many areas of scientific inquiry, including archaeology. And I hope you're all excited to try to use it and employ it in your own research. So if you have any questions, I would love to answer those. If you want to follow us on Blue Sky, there's our Ancient DNA Lab site, my own personal lab, which is Microarch Lab, as well as my own personal page. And one small shout out to say we're hiring 2 people in this space, including 1 ancient DNA research lab manager and technician. So, if you're interested in doing this work, please sing out. We would love to have you as part of the team. So thanks very much. I look forward to your questions.